Good afternoon, family. I welcome you to Metric Hub. Today we are going to talk about gene mutation and Down syndrome, which are key subject or syllabus components that you always see in our exams that we would always need to understand protein formation, that you understand that amino acids are the building blocks of proteins. During the process of protein formation, we understand and we agree as a class that it is mRNA that has been transcribed during transcription that results in the formation of in the process of protein synthesis or protein formation, where the mRNA is going to be read through the ribosomes, and the order of the codons on the mRNA would define the anticodons, which in turn also provide the specific proteins to be formed. So today we are going to talk about mutation that affects, or how does mutation affect the protein synthesis. So in other words, when mutation occurs, it results in the change of the nucleotide bases that will be found on the mRNA that results in different protein to be formed if ever mutation has occurred. So mutation is basically a change in the sequence of nitrogen bases that are found on the on the mRNA that results in the formation of different proteins being formed during protein synthesis. So would want to look into that. I'm sure that you understand that mRNA is formed from DNA as it assimilates during the process of transcription. Just to go into our questions that we have today, we would quickly want to answer these questions, and then we'll be further explaining them as we go. The question here is that there is a table that shows codons that code for some amino acids, and we have got the mRNA codon, and then we have got the amino acids that are there. Is AGU, which is adenine, uracil, and guanine. As I'm quite sure you know that in RNA, we no longer have thymine, we have got uracil. That is one thing that you are going to understand in terms of the nitrogen bases that are found on RNA versus the ones that are on DNA. So that's the matter. The question is saying a mutation caused a DNA-based triplet to change from CTG to CTA, and then describe the effect of this mutation on the protein to be formed. We are seeing our DNA, question 2.2, question 2.2. The question is saying, describe the effect of this mutation on the protein to be formed. What we are saying is that our DNA-based triplet changed from CTG to CTA. The change was from CTG to CTA. So we are saying CTG changed to CTA. And then in this case, we need to understand to say, we would want to see the mRNA codons that are also being provided there. So in this case, in order for us to understand, we need to give the corresponding codons, the shift of the codons that would therefore define the protein that is going to be formed. So in this case, we are saying our mRNA codons, our mRNA codons, they also changed from, we are saying in this case, it was CTG, therefore our mRNA codon in the first before mutation, it was GAU, is it's CTG, CTG, so it changed from GAC, and then it changed to GAU, guanine G pairs with that guanine cytosine thymine, which is now represented by A there is fine, and then we are saying adenine, and then it has got uracil there. So our codons, our mRNA codons changed also from the mRNA codons changed from GAC to GAU. So the question is saying describe the effect of this mutation on protein to be formed. So on our mRNA codon, our answer would therefore say, we are saying, therefore our answer should say codon GAC on our mRNA code on our mRNA it changed to what? It changed to GAU that you can see here, which you can see here. We said GAC changed to GAU because of CTG changed to CTA. So codon GAC on mRNA code changed to GAU, which both code for the same amino acid, of which both code for the same amino acid, which is in this case we are saying GAU, we are saying GAC here we are saying our GAC is this one. Our GAC is that one, which is aspartic acid. It changed to GAU, which is also this one. So what we are saying is that the effect of the mutation is that codon GAC on mRNA changed to GAU, of which both code for the same amino acid, which is aspartic acid. Well, amino acid, sorry, change code for the same amino acid, aspartic acid. I'm sure that we can see we are saying GAC changed to GAU, which is on the table there in terms of the amino acid that is being produced. And don't forget that the amino acids are the building blocks of what? Of proteins. So therefore, there is no effect on the protein being formed in this case. So the effect of the mutation is that there is no resulting in no effect on the protein being formed. Resulting in the same protein being formed. So we are saying the effect of the there is no effect on this mutation, as you can see there. We said our base triplet on DNA changed from CTG to CTA, which actually affects the codons on mRNA. On our mRNA, which we are saying our mRNA is this one. Our mRNA is like that, and then this is our base triplet on what? On the DNA. So there is no effect on the proteins to be formed because they are the codons that are on the mRNA are coding for the same protein. Hence, the same protein aspartic acid is formed. So the same protein aspartic acid is formed. Let's go to the next question, 2.3. Question 2.3. Tabulate the difference between DNA and RNA nucleotides. Tabulate the difference. When we are saying tabulating, I'm sure you understand that tabulating is to say you are supposed to draw a table. 2.3. Question 2.3. When we are saying give tabulating, it is just saying draw a table where we can say DNA, and then we can put a block there, and then we say tabulate between DNA and RNA. And then we are saying do their differences. What is their differences? I'm sure you know that nucleotides are the building blocks of what? Nucleotides are the building blocks of nucleotides are the building blocks of DNA and RNA. So the question wants us to give the differences in these two scenarios. So in first question or first difference one, this one we are saying DNA has got deoxyribose sugar. We are saying it contains deoxyribose sugar DNA. It contains deoxyribose. It contains deoxyribose sugar, and this one also we are saying it has got ribose sugar. The sugar in DNA and RNA is different. DNA, it has got deoxyribose sugar, and RNA has got what? It has got ribose sugar. That is one difference there. And then another difference, which is number 2. Next difference, which we can say DNA, it contains thymine nitrogen base. It has got thymine nitrogen base. Number this one on this side, it has got uracil instead, uracil base. So DNA, it has got the nitrogen bases, which are all of the same, as to say we have got DNA, we have got adenine, we have got thymine, we have got cytosine, we have got guanine. However, in RNA, we have got RNA, it contains what? It contains uracil. It contains uracil. And then one other difference is that the nucleotides in DNA results in the nucleotides in DNA results in a double strand structure. The nucleotides, we're saying, forms a double helix structure. And then this one, it forms a single strand. RNA forms a single strand. DNA has got a double strand. So the differences are such as those. We are saying DNA has got a double helix structure, and RNA has got a single strand, which is the key differences that are there. Those are the key differences, which I'm sure that you remember. If we can further look into that, just to make it clear in this regard, I'm sure that you know that I'm sure that you understand that the structures of these nucleotides of nucleic acid, they are different in that regard, that the other one is a double helix, the other one is a single strand. So in that case, we should understand and make sure that we don't mix or we don't vice versa the two. So I just want to just add some content on this one. The structure, the structure of, we just said the structure of nucleotides. Because certain times again, it comes whereby you are asked to just explain it. It comes where you are supposed to give, or you are supposed to explain, or you are supposed to identify it, and then it will be a problem again, which we are saying we must also just be able to understand this structure to say, how does it look like? So in most cases, we are supposed to know that the DNA structure is made up of 3 nucleotide bases or 3 nucleotide components, which we are saying in this block, we can have P, and then we can also have the deoxyribose sugar there. We've got deoxyribose sugar, and then which is this one, and then this, we can have the nitrogen base, there, the nitrogen base, and then is the last, which is the circle below there, we are saying is the phosphate group. So here I'm just further explaining to say the structure of the nucleotides is like this. That's how the structure is like. So in this regard, let us just quickly just refer back to say, because DNA is a double helix, because it's a double helix, what are we saying? The structure of DNA is a double helix, which means to say that it looks like this. It looks like this casing. It looks like this simple diagram that I'm trying to draw here, which we can have that and that. These are called the nitrogen bases, which are joined together by weak hydrogen bonds that are in between the nitrogen bases. So what is happening is that in this case, we are saying we have got complementary bases that are found there. Complementary bases in the DNA structure, in DNA. So what happens is that we are saying G, it pairs with what? With the C, which we are saying G is guanine and C is cytosine. And then we have got also thymine, it pairs with what? With A, which we can give as to say, I can give this in this scenario as to say, I can just maybe illustrate them by saying in this 1 and 2, we are saying 1 and 2 are complementary bases that can be found on a DNA structure. So we can say it can be either this one, it can be this, which is representing to say 1 is guanine. We can say 1 is guanine there, and then 2 is cytosine, as in like that case. And then in between them, in between here, the complementary bases here, there is a link that supports or that combine the corresponding the complementary bases are joined together by what we call weak hydrogen bonds. Weak hydrogen bonds. The weak hydrogen bonds are found in between the bases there. They are found in between complementary bases. So likewise, we are saying these bases, we are saying they are subtended by a weak hydrogen bond that joins them together there. They are joined together by a weak hydrogen bond that is there, which we are saying it is weak in order to allow for DNA replication, which is another subject, of course, to discuss and talk about. And then I'm sure that we are saying the nucleotides, they are forming this. And then one other thing is that the strand of this, the strands are formed by the strands, the DNA strands. The DNA strands are formed by what? By phosphate sugar molecules. So we are saying phosphate sugar molecules, they do form the DNA strands. The ones which forms the DNA strands. The strands are formed by the by those what we call phosphate and sugar molecules. They are the ones which are forming these strands that we are talking about. The strands are formed by the by the phosphate sugar molecules. Hence, we are saying that is the structure. And then we can also similarly just illustrate a simple structure of RNA to say RNA can be just like that, which we are saying it is a single strand. It is no, we are saying that is RNA. And if you can see there, it is just a single strand. And then as you can see there, it also have its nitrogen bases, which it can have guanine, it can have cytosine, it can have adenine. However, instead of the other one, which is thymine, it has got uracil. So we can have these bases like that. We can have those bases like that. And then, of course, you can see this is a DNA structure. That is a DNA structure here. And then this is an RNA structure, which is a single strand. And then that one is what is a double strand. Was just to bring to attention of the class to say that the DNA structure and the RNA structure, complementary bases are found in DNA, and then in RNA, we only have a single strand, which has got, of course, also uracil instead of thymine, which you see here that we have got uracil here, and then here it is the one that has been substituting. It's thymine that has been, that is substituted by what? By uracil. So that's the thing. That's the idea, or that's the point in terms of the structures. We can further move into our questions. We can move on to the next question. The next question is question 2. The next question is 2.41. Question 2.41, which is saying Down syndrome is a result of an individual having an extra copy of chromosome 21, two genetic variations that can cause Down syndrome are trisomy 21, where we are saying all the somatic cells in an individual have 3 copies of chromosome 21 due to abnormal process that occurs during gamete formation or gamete production. And then we are saying again that two genetic variations that can cause Down syndrome are trisomy 21, and also there is what is called Mosaic syndrome. So in this case, we are saying in trisomy 21, all the somatic cells in an individual have 3 copies of chromosome 21 due to abnormal meiosis that we have identified there. In terms of mosaic syndrome, the individual has only some cells in an extra, with an extra copy of chromosome 21, which is caused by abnormal process during cell division after fertilization. So the question is saying, first question is saying, name the type of mutation that leads to Down syndrome. Name the type of mutation that leads to Down syndrome. The type of mutation that leads to Down syndrome, we are saying it's chromosomal mutation, which is question 2.41. Question 2.41. Question 2.4.1. Name the type of mutation that leads to trisomy 21, whereas type of mutation is chromosomal mutation. Chromosomal mutation. Chromosomal mutation, as you can see, we are saying chromosomes are the ones which fail to separate during the process of meiosis or during the process of gamete formation. Hence, we are saying it is chromosomal mutation 2.41. And then next question is saying abnormal process during gamete production that leads to 3 copies of chromosome 21. Name the abnormal process that leads to that, which is 3. This is A. And then we are saying B. B, what is happening there? The B question, we are saying name the type of mutation that, the type of abnormal process that occurs during the process of this. Question B, what we are saying is that it is called non-disjunction. How do we have non-disjunction? Or we can also call it non-separation. We can also call it non-separation. So what is happening is chromosomes are failing to separate during their process chromosomes are failing to separate, which is causing us to have 3 copies of what? 3 copies of chromosome 21. So it is non-separation of chromosomes or non-disjunction of chromosomes that leads to having 3 copies of chromosome 21, which we are saying a non-separated chromosome, which is 2N, will therefore fertilize or fuse with another N, which would give us a 3N cell. That is the trisomy condition being defined and being referred to here. And then question C, type of cell division that occurs after fertilization. When cells have been formed, what type of cell division that allows growth? Here we are just being asked to understand that we have got what is called mitosis. Mitosis is the process by which cells multiply in order to grow or in order to replace the dead cells. So during fertilization or after fertilization, a zygote is formed, which would further divide and multiply in cells that would result in that zygote developing and growing embryo fetus until a baby is born. So the process of growth that takes place after fertilization is mitosis. Just take note of that. Meiosis, it happens to form this to form the gametes. When the gamete has been fertilized, it grows by a process of fertilization. That's one key system that we need to understand. And then question 2.4.2, question 2.4.2, it wants us to say 2.4.2, describe how the process in question leads to trisomy 21. It is already there. Describe how the condition leads to trisomy 21. Number 2.4.2, 2.4.2. What we are saying, chromosome, what happens there, I can just list the stages here, which is saying how do we have trisomy 21. One chromosome pair 21 failed to separate during anaphase 1 or 2. That is what happens there. We are saying chromosome 21 failed to separate during anaphase 1 or anaphase 2. It failed to separate during anaphase 1 or anaphase 2, and then resulting in a daughter cell having an extra chromosome, where we are saying a daughter cell is formed. A daughter cell with an extra chromosome is formed. Which we are saying, in this case, we are saying this is a daughter cell, which is 2N. A 2N daughter cell is formed instead of having N, instead of having any chromosomes. 2N instead of N. Any, you know, that's to say N must be the normal, it should be the normal. But this cell, we are saying chromosome 21 failed to separate during anaphase 1 or 2. That would result in a daughter cell with an extra chromosome being formed. Chromosome is formed, which is 2N instead of what? 2N instead of N, which is 2N instead of N here. Which is 2N instead of N. So in that regard, we are saying fusion of the gamete with normal chromosomes result in a zygote with extra chromosomes. So what is happening is that that daughter cell that is being formed, that daughter cell which is formed, we are saying fusion or fertilization, fusion or fertilization with normal cell will happen. So in this case, you should understand to say that this is whereby we are saying the process is therefore resulting in a situation where we are saying it is a 2N daughter cell plus a normal cell, 2n total cell plus a normal cell, which is going to give us 3n, or else it was going to be like saying 24 chromosomes is fusing together with what? With 23 chromosomes. In this case, which will result in 47 chromosomes. So in that case, what we are saying is that this non-separation of a cell or processes of non-disjunction that took place in anaphase 1 or 2 resulting in what? Resulting in a trisomy condition. Resulting in a trisomy condition. In a trisomy condition. So basically, that is what we are saying. That is exactly what we are saying. That there is a non-separation of chromosome that took place during anaphase 1 or 2, resulting in a daughter cell with extra chromosomes being formed, which we are saying the daughter cell, it is having 2n, this one, 2n, it is having 2n instead of having what? Instead of having n, which we are saying 2n is abnormal and then the normal is n. So fusion, which we are saying that it is the fusion or fertilization with a normal cell will take place, where 2n plus n is equal to 3n or 2 or 24, which is that one with 2n, 24 plus this normal one, which is n, gives us 47, resulting in a trisomy condition. Then that's what is happening there in terms of 2.41. And the next question is saying, describe two differences between trisomy 21 and Mosaic Down syndrome. Two differences between the two. Describe two differences. Question 2.43. Question 2.43, describe two differences between trisomy 21 and Mosaic syndrome. So the difference is 2.43. 2.43, what are the differences that are there? We are saying one, we are saying trisomy 21, there is an extra chromosome or 3 copies of chromosome 21 in trisomy 21. There is an extra chromosome we have got an extra chromosome 21 or 3 copies of chromosome 21. 3 copies of chromosome 21. So then what is that in each somatic cell, which is found in each somatic cell? In each somatic cell, we have got a cell which has got trisomy 21, while in terms of this, what is this Mosaic syndrome? Mosaic syndrome, there is extra chromosome on some of the cells. There is an extra chromosome. The extra chromosome is found on some of the cells, not each of them. some of the cells. In some of the cells. That is also one point there. And then we can also give to say, if you have seen the question of the scenario is saying trisomy 21 occurs during meiosis before fertilization, but mosaic Down syndrome occurs during mitosis. Trisomy 21, it occurs during meiosis before fertilization. Or mosaic syndrome. Mosaic syndrome, it also occurs during mitosis, after fertilization. So those are the differences. First difference is that in trisomy 21, there is an extra copy of chromosome 21 or 3 copies of chromosome 21 in each somatic cell. Where mosaic syndrome, there is an extra chromosome on some of the cells, not all of them. And then trisomy 21 occurs during meiosis, which is before fertilization, while mosaic syndrome, it occurs during mitosis after fertilization is occurred. So we can say those are the questions, or that's what we mean by those questions there. Let us just quickly finish the last question there, which is our last part of this. One. We can just quickly finish this as to say, the question wants us to say, we need to understand that mosaic syndrome and what, and at trisomy 21, there are different conditions that we must get to know to say that this one occurs before meiosis and the other one occurs after meiosis. That is what is happening there. That is what happens there. So I thank you so much, and I hope that we are going to get a good understanding of these questions, and they are going to guide you further in terms of understanding of the whole concepts and stuff that we are going to be meeting in your exams. Thank you so much. All the best. Please just look forward. Don't forget to follow the channel so that you can be able to continuously get yourself updated with the information. Thank you so much, and have a beautiful day. I thank you. Have a good day and all the best in your studying.